And welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about the history of medical microbiology. So in this particular lecture we will see what is the history of medical microbiology, from where this medical microbiology came into existence, who all are the scientists who contributed their work in medical microbiology, and what is the basic definition of medical microbiology. So let's get started with the topic, that is history of medical microbiology. So the introduction part of the topic says microbiology is the study of living organisms of microscopic size. So we have discussed this in our previous lectures of microbiology, that is what is microbiology. So microbiology is a science that deals with the study of microorganisms, those living organisms that are so tiny that you cannot see with your naked eyes. So you need certain types of microscope to visualize them. So the term was introduced by a French chemist, that is Louis Pasteur. So Louis Pasteur is the French chemist that coined this term, that is a microbiology. So another scientist, that is Antony von Leeuwenhoek, in 1632 designed a microscope, single lens microscope, and demonstrated a little agent of disease which he has designed as animalcules, and these animalcules are now well established entities belonging to the bacteria, viruses, and other pathogens. The organisms being visible to the naked eyes are known as microorganisms. For many years, it was believed that microorganisms arose from dead, especially decomposed organic matter. This was known as the theory of spontaneous generations. So, in this what we have covered so far, that Antony von Hook is a scientist who designed a single lens microscope, and he demonstrated that there is something, but later on you can say established that this disease is caused through bacteria, viruses, and other pathogens. So the organisms that are visualized under this microorganisms, and the organism that you can see under the microscopes is known as they are known as microorganisms. So for many years, students, it is believed that microorganisms that you are seeing under the microscope is these arises from dead and especially a decomposed organic material, and this is known as the spontaneous generation theory. Now the second contribution was done by of Louis Pasteur. So Louis Pasteur in 1822 to 1895, he is a French chemist, generated a strong evidence to show that the microorganism did not arise de novo or spontaneously in the media, but were introduced them from without. So Pasteur also shows that these organisms were maximum in dusty air of towns and minimum in the air of hilly areas where human habitat did not exist. Pasteur also shows that microorganisms were inactivated through the first is boiling at a temperature of 120 degrees centigrade under pressure, that is autoclaved, and at 170 degrees centigrade, that is known as hot air oven. So Louis Pasteur, he was a French chemist, right? So he gives a strong evidence that this microorganism does not exist through spontaneous generation, that is, or they does not exist on a dead or decaying matter, but they are you can say they are present in the dusty air those air that is found in the atmosphere, right? And but he says that these microorganisms are present everywhere, but the number of the number is more in the town area, as well as compared to the number that is present in the hilly area. So where the human beings lives in a town area, so if you take the sample of air of the area that is in the town area, so here microorganisms are present more, but as you compare them to the hilly area where the population is limited, so there the you can say the number of microorganisms are very less. Apart from this, he also says that these microorganisms can also be inactivated, right? How they can be inactivated? By by boiling them, right, at a certain temperature, or you can say, or you can also autoclave them at a temperature of 120 degrees centigrade, right, but under a certain pressure, that is known as autoclave. You can also you can also inactivate them under a temperature that is 170 degrees centigrade, that is a hot air oven. So under these conditions microorganisms can also be, they can also inactivated, right? Now moving to the next, that is the contribution of Robert Koch. So Robert Koch is the first first contribution to the science was demonstration of the character and mode of growth of consecutive Bacillus anthrax. In 1882, Koch discovered tubercle bacillus, and in 1883 the cholera vibrio. For his manifold discoveries in bacteriology, Koch is considered as a father of bacteriology. So Koch, he is also a scientist. So he makes a different contribution in discovery of tubercle bacillus in 1883, that is the cholera vibrio. He also made discoveries in bacteriology, that is the reason he is known as the father of bacteriology. So in exams or in MCQs or in interviews, generally this question arises. So who is the father of bacteriology? So Robert Koch is the father of bacteriology. The first what he has discovered the tubercle bacillus, and in year 1883 he also discovered the cholera vibrio, and he has makes a magnificent, you can say, discoveries in bacteriology, and that is the reason he is known as the father of bacteriology. So what are the postulates about of Koch? So to confirm the claim that microorganisms isolated from a disease was indeed and the cause of this, Koch postulate a set of criteria. So according to this postulate, microorganisms can be accepted as a consecutive agent of an infectious diseases, and only if following are satisfied. So the first is the isolate should be found in every case of the diseases and under the condition which explain the pathological changes and clinical features. It should be possible to isolate the conjugative agent in pure culture from the lesion. When such pure culture is inoculated in appropriate laboratory animal, the lesion of the disease should be reproduced. So he makes certain postulate. According to these postulate, so the microorganisms, they can be accepted as a consecutive agent of any infectious diseases. So unless and until they microorganism who is to whom they are मतलब कि जब तक ये माइक्रो ऑर्गेनिज्म को आप एक्सेप्ट नहीं करोगे कि ये सारे जो भी पोस्टुलेट हैं, जब तक कि वो कॉन्जिक्यूटिव एजेंट्स नहीं कहा जाएगा. तो what are the different consecutive, what are the postulates? So he say the microorganisms should isolate, the isolate should be, you can say the isolate of microorganisms should be found in every case of the diseases, whether under the condition which the pathological changes and the clinical features are explained. They should be possible to it should be possible to isolate the consecutive agent in a pure culture from the lesion, and after isolated this conjugative agent in the pure culture, they are properly inoculated in the laboratory, and the lesion of the diseases that should be reproduced. Now moving to the next, that is antimicrobials. So what are antimicrobials? So antimicrobials are the therapeutic substances that should be prevent or treat infections. They include antiseptics, antibiotics, antivirals, and antifungals and antiparasitics. Disinfectants are antimicrobial agents applied to a non-living surface. Antimicrobials can kill microorganisms and or to prevent their growth by targeting key steps in cellular metabolism, such as synthesis of biological micromolecules, the activity of cellular enzyme, or the cellular structure such as cell wall and cell membrane. The presence of antimicrobial agents in the ecosystems, whether a natural or a man-made, always has an ecological impact. So this antimicrobial. So what are antimicrobials? So antimicrobial is nothing but it is a therapeutic, you can say, a substance. And they are used to treat, or you can say prevent, any infections. So they are basically a substance, therapeutic. They have a therapeutic properties in them, and they are the substance that is used to prevent, or you can say treat, any infectious diseases. So they include antiseptics, they include antibiotics, they include antivirals and antifungals and antiparasitics. So these disinfectants are antimicrobial agents applied to a non-living surface. Then this antimicrobial, so what they actually do, they kill the microorganism, or you can say they prevent the growth of microorganism. How? By targeting prevent their growth by targeting the key steps in the cellular metabolism, such as synthesis of biological macromolecules, the activities of cellular enzyme or cellular structures such as cell wall and cell membrane. So the presence of this antimicrobial agent in an ecosystem, whether natural or man-made, are always have an ecological impact. Now moving to the interviews based question. But before moving to the interview based question, let us see what all we have completed so far. So firstly we have completed about you can say we have studied about the history of medical microbiology. So who is the scientist who coined the term, that is of medical microbiology, or you can say microorganisms. Then what is the contribution of Antony von Leeuwenhoek? What is the spontaneous generation? So after that we have seen the contribution of Louis Pasteur. We have seen how the microorganisms are inactivated. We have seen the contribution of Robert Koch, the postulates of Robert Koch, and we have seen what are antimicrobials. Now let's discuss what are the interview questions that is important for you to go through. So these are certain important questions that are going to ask in the interview. You can just simply take a screenshot of this. So the first question says, what is a spontaneous generation? So we have studied about this. Spontaneous generation says that microorganisms, they are generally or mainly present on the dead decaying surface, mainly on the decomposed surface, and this is known as the spontaneous generation, and from where the existence of microorganisms came into existence. Then comes what state the contribution of Louis Pasteur. So this question is very important. What are the contribution of Louis Pasteur? So that we have already studied that Louis Pasteur stated that and he gives the strong evidence, right, that this microorganism did not arise spontaneously. They did not arise through de novo or spontaneously in the media, but they are introduced, were introduced without. And he says that the microorganisms were maximum in the dusty air of the towns as compared to the microorganisms that are present in the hilly areas, right? So he also shows that how the microorganisms are inactivated through boiling, boiling at a different degree temperature and different pressure, right? So this is the contribution of Louis Pasteur. Then who is considered as the father of bacteriology? So Robert Koch is considered as a father of bacteriology. Why? Because he has discovered manifold discoveries in bacteriology by discovering tubercle bacillus. He also discovered the cholera vibrio, right, in year 1883. So by making a huge discoveries in bacteriology, he is considered as a father of bacteriology. Then the question says then the question says, what are antimicrobials, right? So what are antimicrobials? So antimicrobial is nothing but it is a therapeutic agent that is used to treat infectious diseases. So antimicrobial agents, antimicrobials includes antiseptics, antibiotics, antivirals, antifungals, and antiparasitics. And what is disinfectants? So disinfectants, they are also antimicrobial agent, but they are applied on the non-living surface. So how they kill antimicrobial? So antimicrobial, they can kill microorganisms, or they can also prevent their growth. How? By targeting the key steps in the cellular metabolism, such as synthesis of biological macromolecules, the activities of cellular metabolisms, cellular enzymes, or you can say or cellular structures such as the cell wall, cell membrane, right? So then comes the last, that how microorganisms are inactivated, again by boiling, boiling at under a pressure at a temperature of 120 degrees centigrade with a pressure and a certain pressure that is known as autoclave, by boiling at a degree of 170 degree, that is a hot air oven. So under a high pressure, under the very high temperature, microorganisms can be inactivated. So microorganisms need a very favorable conditions to survive. So when this condition is not there, so microorganism does not survive, cannot survive, and at a very high or a harsh temperature they get inactivated, right? That is the reason why our mothers boil milk, that is known as pasteurization. Why? Because at certain degree temperature, microorganisms that are present in the dairy product, especially in milk, they get inactivated, right? So this is all about the history of medical microbiology. Hope you have understood the lecture. If any doubt, any queries, you can leave your doubts and queries in the comment section below. or in the next lecture I'll be answering all the queries. You can also visit our website, that is www.itlsacademy.com. Here you can get updated with the different courses. You can also call our helpline number, that is 7080833450. So you can directly call on this number to have if you have any queries. And at last, don't forget to subscribe ITLS Academy on YouTube, and you can just simply press the bell icon and get updated on different lectures on different series of medical microbiology, simply microbiology. So thank you students for watching. Please do like, share and subscribe. Thank you.